Hello everybody, this is test 2 from fall 2013. I'm going to go through the answers here, explain why they are what they are. I've already gone through and given slide references to as many of the problems that I can trace back to 1 or 2 slides. Remember, the format is unit, slide number. So the first one you can find in Unit 4, slides 14 and 15. It's talking about which reaction takes place mostly in liver cells. Remember, the liver is what's going to be processing all of the other sugars, fructose, mannose, galactose, primarily. So it's good practice to go through all the other answers and make sure you can identify what's the Krebs cycle, TCA cycle, identify what the PDH complex is, and these are both glycolysis reactions. So glycolysis is going to come up again, so make sure you are really good with those reactions still. This protein is not found in muscle cells. Sucrase, that was from Biochemistry 1. It was also known as invertase. It's the thing that breaks down sucrose into glucose and fructose. So make sure to review your GLUT families. Those come up a lot too. That was somewhere in the middle of Unit 3 in your notes for what the GLUT families do. Number 1 and 3 are in all. GLUT 4 was muscle specific. GLUT 2 was mainly liver. Lactate dehydrogenase is going to be found in muscle cells as well as red blood cells. Hexokinase, all cells. Porin, all cells that have a mitochondria. This molecule is essential for the synthesis of lactose and sucrose. So remember, these both have a glucose in them. Lactose is galactose plus glucose, and sucrose is fructose plus glucose. So glucose is going to be your answer. Lactose only, everything else is lactose only as well. Remember to review lactose synthase. Lactose synthase is composed of this and this. So a 2-part enzyme, that comes up as a question sometimes. Which enzyme is not used in the conversion of fructose to pyruvate in the liver? He likes to drive this point home that when you have fructose, it skips that key regulatory step in glycolysis. So PFK1, it does not touch anything beyond that. So review on this slide where fructose enters the glycolysis pathway, and you'll see all these other enzymes are used. Which enzyme is not used in the glycolytic oxidation of mannose, galactose, and glucose? Don't let the words throw you off. That just means glycolysis. So if we're looking at all of these, fructokinase stands out. That one is for fructose only. Which metabolite is not typically synthesized in liver cells? Liver shouldn't be making any lactate. That should be red blood cells and muscles only. All cells are going to do anything from glycolysis and anything with the mitochondria from the PDH through the Krebs cycle. Palmitate, don't let that throw you off. It's a fatty acid. It's the one he uses all the time to represent fatty acids. It's produced by the liver. So make sure to make note of that. Which molecule is known to bind to lactose synthase? So that threw me off when I read to bind to lactose synthase, but I know that UDP galactose plus glucose turns into lactose via lactose synthase, is the enzyme here. So that's why B is the answer. Definitely review progesterone and prolactin. It's around that slide. I don't think it's on the same one. But this one was about knowing the specific reaction of lactose synthase. Protein appears to be found in all human cells. Remember, anything from glycolysis metabolites or enzymes. Muscles only, mitochondria only, liver only, producing galactose. And this was from Unit 3. This is the example of something that's not found in humans. Usually likes to ask a question about the galactose disease. It's found on slide 20. The answer to that is galactitol, the alcohol version. This reaction is catalyzed by peripheral cells. So we know fructose is usually done in the liver, but in peripheral cells, he's talking about the adipose pathway. So you can review that here. And in that pathway, you're using hexokinase. Fructokinase is the one in the liver, as well as triose kinase, liver as well, for fructose. Glucose only, mannose only. This reaction does not take place in liver cells. This is a pretty common thing he does to show something making sucrose, and immediately you have to think plants, and that's not going to be done by any human cells. Again, we see 3 glycolysis reactions and the PDH complex. Which enzyme is known to be turned on after birth? This lactose synthase. That's just a straight memorizing problem. I think this might be the slide with prolactin and progesterone. Which molecule is not typically found in brain cells? Should be no lactose in the brain. Anything from glycolysis with the Krebs cycle should be, or will be found there as well. Lactose should be found either in your digestive system because you ate lactose, or lactating tissue. So moving on to Unit 5, it's talking about prosthetic groups in the PDH complex. So there's 2 different versions of it on slides 20 or 29. Find which one you like better. I think they have mostly the same information on it. These molecules appears to be the smallest. This is a metabolite versus protein or enzyme. So all these you can immediately cross off because they're enzymes. They're going to be bigger than any metabolite. Porin, membrane transporter. So it's a protein as well. Just know that this is going to be smaller than any sort of metabolites. Are always going to be the smallest one when it comes to a question like this. Which of these metabolites is known to inhibit the PDH complex and reactions 3 and 4 of the Krebs cycle? So make sure you know these 2 slides really well. This is the PDH, and this is the Krebs cycle regulation. Remember, the point of all of this is to make ATP. So if you already have ATP, it's going to slow things down. One of these enzymes uses water as a substrate. This is where it comes in handy to know the full reactions. So the full reactions are on slide 44. There's 2 more that use water as a substrate. Aconitase uses it and produces it. It's a little weird, but fumarase uses it as well. So look out for the other 2 that uses water. He usually asks a question about water in the Krebs cycle, so make sure you look up all the places where it's either produced or used. Which mitochondrial metabolite is not generated through a redox reaction? Redox reactions are going to be your dehydrogenases. So citrate is the answer. PDH complex, remember, PDH is also the dehydrogenase as well, so that's where the acetyl-CoA is coming from. And these are coming from the redox reactions in the Krebs cycle, which is an iron-sulfur enzyme, conatase. If you look on that slide again that lists all the reactions, you'll see that succinate dehydrogenase is an iron-sulfur enzyme as well. Which metabolite is not a net product of the TCA cycle in brain cells? There's no actual ATP produced. Now this gets switched into ATP, but this is a product of oxidative phosphorylation. Which is not a redox enzyme or enzyme class 1. It's going to be your synthetase. From the electron transport chain, complexes 1, 2, 3, and 4 are all going to have redox activity. So anytime you see something like that, we'll see that later when we get to that part of the test. So again, anything dehydrogenase is usually, or always, you can cross those out, those are always going to be redox enzymes. The molecule stimulates the conversion of pyruvate to acetyl-CoA. On the PDH complex regulation again. CoA is the answer. How many carbons of fructose are fully oxidized in the mitochondria? That's going to be 6. We're talking about fully oxidized, so the 6 are going to enter. Fructose is a 6-carbon molecule, so it's going to enter as 2 pyruvates, which each have 3 carbons, and one from each of these are going to be oxidized in the PDH complex, and then the other 4 are going to be in the TCA cycle. So in the whole mitochondria, which contains both of these, the answer is 6. If you change this to TCA cycle only, then the answer would be 4. Or you could word it to somehow, before the Krebs cycle in the mitochondria, then it would be the PDH complex. If you were to say in the cytosol, that would be none. Another regulation question. So this slide has come up quite a few times. What inhibits both things? ATP. The purpose is to make them. This vitamin does not appear to be essential to alpha-ketoglutarate dehydrogenase. Remember, alpha-ketoglutarate dehydrogenase is very similar to the PDH complex, and for that you need vitamin 1, 2, 3, and 5. So let's see, we have 1, 2, 3, 5, 6. So that's the answer. Just making sure I actually hit play and haven't been talking to myself for the last 15 minutes. Good. 26. The TCA cycle metabolite is, which is the most immediate precursor of palmitate. So this is on either the last slide or one of the last slides where it shows the other things the Krebs cycle can make. Always a question on this one. Palmitate is our fatty acid, which comes from citrate. So look through everything. These two can both make glucose. One of them can also make amino acids. Some can make purines. Some can make pyrimidines. He does like this one a lot too. This is the one that can do heme groups or porphyrins. So make sure to know that slide pretty well. Which enzyme would be present in a protein extract from the inner mitochondrial membrane? So So don't let this confuse you. He never says it in class, but it's saying what is membrane bound in the inner mitochondrial membrane, and that is succinate dehydrogenase. So complex 2 of the ETC. This would be from the outer mitochondrial membrane. So moving into Unit 6, definitely know the regulation slide. Of course, always going to be a question from there. So oligomyosin is going to inhibit the ATP synthase. This is a common metabolite for the MAS shuttle found here, or the GPS shuttle found here. They both use NADH. So these slides in particular are just the illustrations, which I find helpful to me to write them out, to visualize it. There's a few slides before and after talk about it as well, so find out whatever works best for you, but definitely know all the enzymes involved and what their purpose is and how they work. The shuttles always come up on questions. Let's see which of these has the highest standard reduction potential. He likes to ask a question from this slide, but I don't know how to memorize it or teach it. Usually it's NADH or NAD+. You can look at maybe the top few and memorize those and hope that's the answer. But that's a tough one. Molecule is not a substrate for oxidative phosphorylation. AMP, ADP would be the answer, or would be one. Everything else is used somewhere in oxidative phosphorylation. You can find that on slide 5 amongst many other slides throughout the unit. That was just the first one I came to that had that on it. This reaction of the electron transport chain takes place in, remember the succinate with FAD, that's going to be complex 2. It's the Krebs cycle link. That's found on multiple slides as well. Alright, so there's usually a question like this. This would take, a whole SI session to kind of go through all the different ways he can word this, about what fraction of it's the one math problem he throws in from the unit. He even messed up on this one because these are both answers on the answer key. Figuring all that out takes a lot of work and a lot of math. If you're really interested, if you have really firm understanding, go to these slides and they're going to give you what you need to figure this one out. I think it's historically been a commonly missed one or commonly thrown out question. Which molecule does have a transporter? I was looking through this. I believe he meant does not have a transporter because NADH is the answer. NADH, that's the whole point of the MAS and GPS shuttles. So there's shuttles for these guys. On this slide, you see shuttles for these two as well, the carriers and translocases. Which reaction is catalyzed by glutathione reductase. That's a pretty complex pathway to know on slide 38, but a question I think almost always comes up from it. So the glutathione reductase is A. All the other ones here listed are going to be found on that same slide. So that's a good one to know. Which enzyme does not belong to enzyme class 1? ATP synthase. All of the oxidative phosphorylation enzymes are going to have redox activity, as well as anything with the dehydrogenase. Alright, so the 3 out of the last 4 have to do with thermogenesis. So make sure to review slides 86 and 87. Which molecule is known to stimulate thermogenesis? It's the fatty acids. I'm going to come back to 38. which is membrane-bound thermogenin. Remember, that's UCP1. It's the same thing. And the uncoupler to oxidative phosphorylation is thermogenin as well. But all these other ones I think are found on that regulation slide, which I think was the slide 90. Make sure you really know that, so we'll probably mix it up quite a bit. There's so many different questions you can ask on the regulation. So back to 38, which enzyme does not appear to have prosthetic groups? All of the complexes in the electron transport chain do. ATP synthase does not have any prosthetic groups. That's all I got for you. Good luck in your test.